Hello everyone, welcome to sqadia.com. I'm your host Sannan Rehman and we're discussing muscular systems today. This is the second section of the presentation, molecular regulation of the muscle development. In this section, we'll be talking about the muscle origins, from where these muscles actually originate, the various genes involved, and the patterning of muscles throughout the human body. I hope you're excited about this section. Let's take a look into this into little detail. So, we have various muscular regions within the body. You can divide them broadly into four categories. These are the cervical region, that is the muscle of the head, the thoraco-abdominal region, the muscles of the torso, that is the thorax and the abdomen, the upper limb, the upper appendage, and the lower limb, that is the lower appendage. So we talked about two domains within the body from which the muscles originate, the prime axial domain and the abaxial domain. I'm not going into the detail of these as these have been discussed in the previous section. If you're not clear on these, go to the previous section, listen to them once again so that you know how the prime axial and the abaxial domains are formed. So let's see which muscles originate in the cervical region from the prime axial and the abaxial domains. So first of all, the cervical region, we have the scalene muscles and the genohyoid muscles. They originate from the prime axial domain, and only the infrahyoid muscle originates from the abaxial domain. Now we have the thoraco-abdominal region. In this, all the intercostal muscles, they originate from the prime axial domain. However, the abaxial domain gives rise to the pectoralis major, pectoralis minor, the external oblique, the internal oblique, the transversus abdominis muscles. These are the muscles of anterior abdominal wall. The two obliques are the transversus abdominis. Pectoralis muscles are the muscles of the anterior chest. Transversus abdominis, again, muscle of the anterior abdominal wall. Sternalis, the muscle of sternum. The rectus abdominis, the central abdominal muscle, and the pelvic diaphragm. Now, the muscles of upper limb that originate from the prime axial domain, these are the rhomboids, the levator scapulae, and the latissimus dorsi. Now, all the distal limb muscles, they originate from the abaxial domain. And muscles of the lower limb, all of the lower limb muscles, they originate from the abaxial domain. This is a very important slide which gives you an idea how the muscular development throughout the body takes place. I'll recommend you to try to memorize it so that you have a clear picture of what happens during development in your mind. Now we take a look at various genes involved in the process of muscular development. So we have bone morphogenic protein, BMP4, fibroblast growth factors, from the lateral plate mesoderm. We have WNT proteins. These are from the adjacent ectoderm. Together, they act on the ventrolateral dermomyotome. We talked about the dermomyotome before. The ventrolateral lip was where the muscular elements were being formed in a somoid. This expresses the muscle-specific gene, which is known as MYOT. So, what happened? We had two genes, bone morphogenic proteins 4 and fibroblast growth factors. Where did these come from? From the lateral plate mesoderm. These were acting along with the WNT proteins from the adjacent ectoderm. Together, they acted on the ventrolateral dermomyotome to express the muscle-specific gene MyoD. This is the most important step in development of muscle in human body, in the human fetus. If anything goes wrong with the expression of genes involved in this process, various types of anomalies or absence or complete or partial absence of muscles may be seen. So this BMP4 induces WNT proteins by the neural tube. It is happening at the same time as low concentration of sonic hedgehog proteins reach the dorsomedial dermomyotome. Together, this BMP and the low concentration of sonic hedgehog genes induce the expression of MYF5 and MYOD in these cells. So these are the names of the genes which have to be taken as such. They're not acronyms or do not stand for anything. These are simply the names of the genes. So both are the members of transcription factor family, myogenic regulatory factors. Myogenic is muscle, regulatory factor is something that regulates or controls it. These activate the pathways for muscle development. So what happens is all of these genes initially they act on the muscle, somite. This somite induces the formation of gene MyoD. This MyoD, under the influence of BMP4 and sonic hedgehog gene, tends to produce or increases the expression of MYF5 in the cells. These both MyoD and MYF5 are members of the myogenic regulatory factors family of transcription factors. This activates the pathways for development of muscles. So muscle development is a very complex pathway in any teratogen that may act during this may result in a faulty development of muscles or partial or complete absence, as whatever the case may be. So patterning of muscles, it is controlled by the connective tissue into which the myoblasts are migrating. So the patterning is controlled by the connective tissue into which the myoblast, the precursor muscle cell or the adult muscle cell, is migrating. The neural crest cells form these connective tissues in the head regions. The origins of the connective tissue in the head regions are the neural crest cells. In the cervical and the occipital region, they differentiate from the somatic mesoderm. So in the cervical and the occipital region, the origins of these connective tissue cells is somatic mesoderm. In body and limbs, they originate from the parietal layer of the lateral plate mesoderm. So very evident from the statement, in the limbs and the body, the torso, they originate from the parietal layer of lateral plate mesoderm. So this was how the muscles originated, the muscles pattern throughout the body. The patterning of muscle is very important. However, since they are extensive throughout the body, the complete or partial absence of one or more muscle may not be that evident as you might think. It's very common. Not very common, it's common to see people with muscle deficiencies. However, a pronounced muscle deficiency is only produced when there is a massive gene misexpression or a teratogen which acts in the way of the gene expression and induces a major change. Let's hope let's hope that you were able to get this into your minds, because the clinical correlates are very important and that would be very helpful when you will come across them. Thank you. Keep watching sqadia.com.